Now, last time we were looking at how point mutations are classified. And so I'm going to give you some examples about their classification and how that works. So we're talking about the three major types. We've got base substitutions, where we have an incorrect base substituted for a correct one. We have what's called an addition, which simply is an insertion of a base or a couple bases. And then what's called a deletion, which is the removal of a base or a couple of bases. All right. We also talked about how we can subdivide those into some other classifications. And I'm going to give you examples of each one of these as we go along. So we're going to go and begin with a little DNA segment. To me, when I look at a mutation, not only do I go and put what the DNA sequence is, but I will often go and put in what the amino acids are. We'll find out that a lot of these classifications are based on their effect on the amino acid sequence coded for by the change. So let's begin with our first one here. And we're going to begin with a simple one. Here is my originally unmutated sequence. As you can see, we have 5 codons. Right? This is what the transcript would look like, and this is what the genetic code translates those amino acids into. So we're going to look at this particular codon right here. As you can see, we have a GGC. And if you look at the mutation down here, this is a GGT. Now, what you're looking at is an example of a base substitution, where we've had an incorrect base, a T, has replaced the normal C. Right? So hence, the general classification is this is called a base substitution. Now, this is also classified as a silent mutation, because if we look at the amino acid that GGC codes for, you'll notice that the mutant codes for exactly the same amino acid. Right? Since there's no change in the amino acid sequence, this is why we call it silent. A silent mutation is a mutation that results in exactly the same amino acid sequence. Right? Now, this happens quite often. In fact, oftentimes the only time that we see a silent mutation is if we look at the DNA and go, hey, look, there's just one little change. And this is most common here in the third base, because the third base is often the one with the wobble, the variation, that there are a lot of codons that will code for the same amino acid, and the difference will be in the third base. All right. So this is an example of what we call a base substitution silent mutation. Base substitution silent mutation. This is how we would classify this one. Oh, that's just to show where the change was? Yeah.
Why is it that DNA changes to a T? Oh, wait,
Yeah, that's the mutant. That's the original. Yeah, it's just a change. Okay, let's try another one. In this case, we're going to use the same codon, but in this case, I've actually changed the first base in that codon. So instead of GGC, as we've seen before, there has been a base substitution. So now we have an AGC. Right? So we have substituted an incorrect base for a correct base. Now, the cool thing about this is look what it did to the amino acid. That codes for glycine. That one codes for serine. Right? So, we call this a base substitution, and you'll notice that we call it a missense. A missense mutation is a mutation that results in a different amino acid. Right? So in this case, we get a different amino acid. So silent, we get the same amino acid. Missense, we get a different amino acid. Now there's one point left, and you'll notice that we have one more subclass to talk about, and that's what we call a transition. Right. Now if we look, go ahead.
What was the one before that? It was a base substitution, but I forgot to get that.
Base substitution, silent. Yeah, base substitution, silent. Now if we look at this one here, we can get a little bit more clarifying by calling it a transition or a transversion. All right, those are both types of missense mutations. All right, the difference has to do with whether you're going from a purine to a purine, or a pyrimidine to a pyrimidine, or a pyrimidine to a purine, or a purine to a pyrimidine. All right, that's a little confusing. But the basic idea is, since we're going from a purine, G is a purine, to an adenine, which is also a purine, that's what we call a transition. So transition mutations occur when we have, for example, a purine gets substituted for your purine, or a pyrimidine gets substituted for a pyrimidine. Now the opposite of that is what we call a transversion. And in that case, we're going to have a purine transferring into a pyrimidine, or a pyrimidine. If I can spell today, that would help. To a purine. All right. So transition, purine to purine, that would be, in this case, G to C, or excuse me, G to A. All right. If, for example, we had a G here and it went to a C, that would be what we call a transversion. All right. Yes? Sure.
So, transition has no change?
It does. It has no change. That's right. So it stays a purine or a pyrimidine.
How is this one a transition?
It's a transition because guanine is a purine and adenine is a purine. Remember, A and G are purines because they're like silver, pure silver. And C and T are pyrimidines.
One has two rings.
Purines. Purines have two rings. Okay. Now the important thing is to go and take a look at the effect on the protein. Sometimes we can get a change in the amino acid sequence and the conformation of the protein stays exactly the same. Or it works just as well. It's equivalent. And let's say that's what happened with this one. Let's imagine that this little protein I'm making is a receptor protein. So we have this little receptor here. We have a little reactant that goes and binds to the receptor. And even though our little mutation may change the conformation of the protein, in this case, the protein still works. And so in this case, we can say that even though we had a mutation, that the proteins are still acting the same. Even though we changed an amino acid, it had no effect on the function of the protein. Now sometimes that can actually go and, let me grab the other one because that one, there we go. Okay. So let's try another one. So here we have our codon, GGC. All right. And in this case, I've replaced the G with an A. All right. Now, this one also goes and is a base substitution because we went from one base to another base. And it also goes and codes for a slightly different amino acid. Instead of glycine here, it goes and codes for asparagine. Right? So, because of that, it creates a missense mutation. And since we are going from a purine to a purine, this is also a transition mutation. Now, let's say this is a vital amino acid. That this vital amino acid, right, has a big influence on how the protein folds. So we end up with a protein where it's folded in such a way that we've lost the receptor site. So this type of mutation, all right, just one base changing one amino acid changes the shape of the protein and we've lost the function. Right? And this often happens. We'll take a look at some real examples a little bit later. But sometimes little changes like that will have a big influence on what the protein does.
Is that little red dot with the white one, is that the active site over there?
Oh, this is the active site. I just made this as the active site.
I'm talking about the one on the bottom.
Oh, this one? You can see how the active site has moved.
Look at the other one. On the bottom. Is that supposed to be like a little bit separated?
No, I've actually gone and I tried to show that it folded differently, so we've lost the active site. All right. Now, so we've looked at base substitution missense. Let's take a look at a really cool one that's called a nonsense mutation. In this case, what we have is a premature stop. Right? So here we have AAG, and you'll notice that we have a base substitution that goes from A to a T. So we've substituted T incorrectly for an A. And if you look, it codes for UAG. If you look up on the genetic code, that's going to be a stop. So we end up getting a premature stop. Here's the normal stop. Here's the premature stop. Now, we call this a nonsense mutation. That's what nonsense mutations do. They code for premature stops. Now, the reason that we call them nonsense, look at the rest of the gene. Right? Are these two going to be translated? These two codons? No. They might as well be nonsense. Right? None of these are going to be translated. So this is why we call it a nonsense. So this one would be classified as a base substitution nonsense mutation. And obviously, this is going to have a big influence on the resulting protein. You're going to get a protein that's going to be much, much shorter. And oftentimes, if you know, short proteins are going to be less complicated. We're going to lose active sites and receptor sites and all kinds of important things. So we end up with, oftentimes, proteins that don't function right because of a nonsense mutation. Okay. Now, we're going to move away from base substitution and start talking about what happens when we have additions and deletions. What happens when we add bases and when we take them away. Right? And one of the interesting things that will happen is what we call a frame shift mutation. Let's talk a little bit about a frame shift mutation. All frame shifts are an addition or deletion of base pairs. The important thing about frame shift is that at the point of a frame shift, right, the sequence, particularly the sequence of amino acids, is going to change dramatically downstream. Frame shift mutations that come from additions and deletions are often the most devastating of all the types of mutations. They can wreak havoc on proteins. So one of the worst things you can do is do some deletions and insertions. They can really change what's happening to a protein. Let me give you a little example. When we think about a DNA sequence, we think about it as a three-letter language. Every word is 3 letters. And the interesting thing about this written language is that unlike ours, where we have spaces between our words, all right, in the genetic language, there are no spaces. Right? Those words are right next to each other. So if you insert or delete any of those letters in the words, you end up getting everything shifting to the left or everything shifting to the right. We say that there's been a shift in the reading frame. A shift in the reading frame. That's why we call it a frame shift. Give you a little example. Let's imagine that our genetic code is like a written language. And so I've sort of made this up. The cat ate the rat. Now, what would happen is if we went and deleted, for example, the C in cat, that would cause every letter to go and shift one space to the left because they are connected to each other. So what was actually gone and part of the third codon is now part of the second. What was part of the fourth codon is now part of the third. Everything has shifted one space to the left. If we have an insertion, everything shifts to the right. We have a frame shift. So if we add an extra C here, this T, which is now in the third codon instead of in the second codon. Right? Right. Exactly right. Now, this happens to us all the time. I don't know about you, but I've had Scantron tests where I've gone and filled in the bubbles and I've gotten one question off, and everything downstream from that is usually wrong. All right? Everything downstream from that is usually wrong. Sometimes if you're typing and you accidentally go and shift your hands to the right, everything that you type on then is just going to be gibberish. All right? So frame shifts are incredibly damaging. They come up with proteins that are weird because the DNA sequence is going to be very strange. Now, they're really easy to identify. What you do, and this is the reason why we go and put the amino acid sequence along with the change in the DNA, because if you can see what the amino acids are doing, then you can say, oh, okay, I know what's happened here. You can find where the change takes place. You just follow the normal sequence until you see an amino acid that's different and keeps being different. And you say, oh, there must be a frame shift in here somewhere. So let me show you a couple of examples of some frame shifts. Here's one where we actually went and had a deletion. So here is a normal codon, TTT. And then you'll notice that in this codon, we go TTG. Where did that G come from? It came from over here. Here's the second G, here's the C, here's the T. Everything has shifted one space to the left. The interesting thing about it, you'll notice that the amino acids stay the same until we get to right here, and they keep being different. If this was base substitution, that would be the same, that would be the same, and so on and so forth. But all of a sudden you get a string of different amino acids. That tells you you've got a frame shift. And you've just got to go and find, are we looking at a deletion or are we looking at an insertion? And this one turns out to be a deletion. So we would classify this one as a deletion, all right, frame shift mutation. Now if you want to put in missense, you sure can, because obviously when we have deletions and insertions, you're often going to get a little missense anyway. So we often just call it deletion frame shift mutation. Let's take a look at another one. This one is an example of an addition. This one's kind of cool. I like this one. Here we have a stop codon, and you'll notice that we've added a C. The T is still there. It's just been shifted one space to the right. So in this case, what's happened is we've had a base pair added. So this is an addition. Now, we've talked about nonsense as a premature stop. What would a sense be? Changes it for an amino acid. Right? So the next stop might be over here, or it might be 1,000 codons down. Basically, what you end up is a protein that's going to be much, much longer. So a sense mutation is when you change a stop codon, all right, to an amino acid. So you end up with a much, much longer protein. And that's why we call it a sense mutation. Right? And as you can see, everything has shifted one space to the right, so we call it a frame shift. Now, there you can have additions and insertions, all right, or excuse me, additions and deletions without having a frame shift. Right? Let's imagine that we went and took out an entire codon. Let's say we removed that one codon right there. Right? Would that have any effect downstream? No. You still have the glycine next. You still have the stop next. If you remove an entire codon, all right, it's not going to change the reading frame. Everything's going to stay the same downstream. Let's say that we went and added an AAA right here. All right? That's not going to change anything downstream. So you can have additions and you can have deletions without a frame shift. So if we kind of look at the general category, if you add or delete a single base, that's going to result in a frame shift. If you add or delete two bases, you're going to also have a frame shift. But if you add 3 in a row, all right, you're going to have an extra amino acid. We get the same exact frame as the original. So we end up in the right frame. If we add 4, if we add 5, if we add 6, not going to be a frame shift. Right? It's going to be in the right frame. So, what that means is you can get some mutations that are additions or deletions that are not going to be frame shift. And we'll look at a really famous one that's an example of one where you have, I won't give it away, but you have an addition or a deletion where you end up with not a frame shift. Alrighty. Okay. Any questions on those base substitutions, base additions, base deletions? All right. Now I want to tell you about some disorders. These disorders generally affect coordination and muscle action in people. And these are some serious genetic disorders. These were quite a mystery for us for a long period of time until we were able to actually go and sequence them. And we found that people who suffered from these had a very unusual sequence of bases that were found near the genes that actually go and cause the affections. And the strange thing is that these are insertions that are repeats of the same little 3 bases over and over and over and over again. Let me show you what I mean by that. These are actually called triplet repeat expansion mutations. Right. In this, for example, if we look at some examples, we'll take a look at Huntington's disease, which is probably the most well-known. We find on chromosome number 4, near a particular gene, people with Huntington's disease actually go and have 40 plus repeats of just CAG, 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 CAG. The interesting thing about them, and this is why it stumped geneticists for so long, is that we find that as we go through families, we find that some members of those families, depending on the generation, will have all of a sudden a greater number of repeats. So one family member might have 40, a couple generations later, someone else might have 42. A couple generations later, somebody else might have 43. They seem to add on as you go. That's why we call it expansion. We think there's a mechanism. In fact, we're pretty much sure that we pinpointed it. It had to do with a mistake that happens during replication. Right? If you remember, when we make the lagging strand, there's that strange little loop that forms. Sometimes there's a little hiccup with that loop. And with the hiccup to that loop, you end up getting an extra little repeat. Now, as you can see, there are quite a few different disorders. You may have heard of Fragile X that usually attacks males since they just have one X chromosome. These are problems with coordination. Huntington's disease is probably the most famous. You have a couple of ataxias. Ataxias is people who have a very difficult time keeping their balance. Instead of putting one foot in front of the other, they often go and walk on a wide stance. We have some muscle wasting going on, and these are all caused by these little random triple expansion repeats. Now, the most famous one is this one right here. This is called Huntington's disease, or HD. The terrible thing about this is people who are born with HD don't develop symptoms usually until middle age, usually about age 40. Right? And what happens is they start to stumble around. They start to have trouble keeping their balance and walking. In fact, it's often described as Huntington's chorea. And if you look at the word chorea, it's C-O-R-E-A, as in choreography. It means dance. So Huntington's dance. And one of the best researchers in the 1980s, a woman by the name of Nancy Wexler, who helped locate the chromosome and the closest gene, the marker where the problem is, her mother died from Huntington's disease. And the family discovered her symptoms first off because she was walking down the sidewalk and was stopped by a police officer who stopped her because he thought she was drunk. All right. She was staggering. It wasn't alcohol. It was the first symptoms of Huntington's disease. All right. Now, Huntington's disease starts destroying a part of the brain called the basal nuclei. In the middle of your brain, there's a little place that helps you coordinate movement called the basal nuclei. And what happens is those neurons start to die. Because it coordinates movement, you start having problems. When we go and walk normally, you notice we swing our arms. A person who has Huntington's chorea first starts having problems controlling the movement of their body. So their arm swings are weird. Right? Over time, it's a slow disease. Sometimes it takes a couple decades to actually go and kill you. So it's progressive. The brain gets worse and worse. Eventually, these poor folks end up in nursing homes. Right? Now, let me show you what the brain looks like of somebody. This is a normal brain, right here. And the basal nuclei are loaded right about there. This is somebody with HD. And you can see the loss of brain tissue. The problem with this is a dominant trait, which means if you have a parent with it, means you have a 50% chance of coming down with the disorder. Now, the reason it's prevalent, right, is because by the time symptoms first appear, most people have already reproduced. So they've already passed it on to their kids. Right? If the symptoms show up around middle age. Now, there's a test for it. And this was developed in the 80s. Right? You can go and tell if you're going to have, if you had a parent with HD, all right, you can find out if you're going to come down with HD or not. All right? The interesting thing is about the age of onset has to do with the number of repeats. So, for example, if you have, for example, let's say 40, you may not get symptoms until you're in your 60s or 70s. But if you have, let's say, 100, you may start showing symptoms in your 20s. Right? In fact, some of our early symptoms are from people in the early teens. So the greater the number of repeats, the earlier the onset of symptoms. Okay. Now, we've gone a little bit through the different types of mutations, the point mutations. So I'm actually going to go and show you 5 real mutations. These cause real diseases, and I want you to identify what the type of mutation is. So on your notes, you may want to number 1 through 5. 1 through 5. I'm going to go through them first, and then we'll come back and we'll see how well you did. So let's begin with our first one. Right. We've talked a lot about sickle cell. Right. So here is the normal sequence, and here is our mutant sequence. Right. So what I want you to do is classify, right, identify the type of mutation this is. Hey! Oh, no worries. We're glad we're here. We're just classifying some mutations. So we've got 5 real mutations that we'll look at, and we're just going to go and see if we can classify them. So here's our first one. Okay, everybody ready for number 2? Now, there is an interesting genetic disorder, and it's called cystic fibrosis. And cystic fibrosis occurs because of a little protein, a channel protein, that helps move chlorine from one side of a cell to another side of the cell. And it turns out that that little protein, that channel protein, doesn't work right. What ends up happening is people with this cystic fibrosis start having huge amounts of mucus fill up their lungs. The mucus spreads, ends up in their digestive tract, and they start having all kinds of problems. This is the most common type of lethal mutation, lethal recessive mutation that we find in people of European heritage. And most of the time, people with this disorder, from the time they're very young, have to go in for respiratory therapy. They're usually placed on medication called pulmazine that goes and breaks down the mucus. They often go and take prophylactic antibiotics to prevent infections. And the life expectancy right now is probably about 40. So this is called cystic fibrosis. Now I mentioned Francis Collins, who is the head of the National Institutes of Health and former head of the Human Genome Project, was actually the researcher. He was a medical geneticist, and he discovered this mutation that is the principal cause of cystic fibrosis. So this is the most common cause. Now it turns out there's about 50 different mutations that cause the same trait. This is one of the more common ones. So take a look at it. So as you can see, there's the normal gene. Whoops. There's the normal gene. And here's the mutant gene. Since this is a recessive trait, if you have two carriers, right, they have a 1 in 4 chance of actually producing a child with cystic fibrosis. This should be, everyone should be tested for this if they plan on having children. If you discover you're a carrier and you discover your potential spouse is a carrier, all right, then you may want to consider another form of reproduction, right, because you have a 1 in 4. I had a student several years ago who had no idea that she was a carrier, and her husband had no idea that he was a carrier, and their first child had cystic fibrosis. So, a good idea to get checked. Okay. Anyone need more time on this one? Okay. This next one is an interesting one. For years, doctors used to go and think that treating disease was simply a matter of bleeding someone, opening up a vein and dropping out half a pint or so of blood. It turns out there really is one disorder where that's beneficial, and it's this one. It's called hemochromatosis. And what it is people with this disease abnormally store iron in their body. And so iron starts being deposited in organs. So the liver starts getting filled with iron. The kidneys start getting loaded with iron. And it can be damaging. They usually discover by accident that when they give blood, they feel great. And so because they feel so great, every month they go and start donating blood. It's only later they discover that, hey, the reason I feel good after giving blood is I have this odd genetic disorder. So there is a treatment, and it's kind of beneficial to humanity in that you just donate a pint of blood a month. And it's a win-win. So let's take a look at the little change here. So here's the, oftentimes when we say normal, we call it wild type. That's the normal gene. And here's the mutant. Anyone need more time with this one? Okay, the next two are ones that produce an abnormal type of hemoglobin. We've talked about how sickle cell produces an abnormal type of hemoglobin. This is also one that produces an abnormal hemoglobin. And it is found endemic in places like Italy and Greece, around the Mediterranean Sea. The symptoms may go from mild to severe, but since you have abnormal hemoglobin, you're going to suffer from anemia. All right. This, because it's located near the Mediterranean Sea, is called thalassemia. Thalassa is Greek for sea, so literally sea blood. Now, as I mentioned before, there are a lot of different mutations that cause the same trait. So in this case, I'm going to show you two variants that both result in thalassemia. Right? So these are two separate mutations. So here's our first one. Right. Anyone need more time on this one? Okay, let's go to the last one, and I've saved the trickiest one for last. This is a second variation. The normal or the wild type is on top. The mutant is down below. All right. Does anyone need more time on this one? All righty. Let's head back to the first one. All right. What type of mutation did we get for this first one? What you got there?
Transversion?
Let's see. Missense transversion. What else you got? What's the general one? We went from an A to a T. We've had the incorrect base instead of the correct one. What's the general way we call that? Base substitution. This is a base substitution. Everything else is right. Base substitution. All right. Gotta have that. Those are the top three. Remember, base substitution, addition, or deletion. Those are the three that you have to have. All right. So this one is a base substitution. You're absolutely right with the other one. This, we changed glutamic acid to valine. So in that case, that's going to be a missense. You can also see where we've gone from an adenine to a thymine. That's a purine, and that is a pyrimidine. So that's transversion. So that one would be classified as base substitution, missense, transversion mutation. Any questions on that one? All right. I love this one. This one is a little tricky, but it's also really easy to identify. This is the one for cystic fibrosis. What did we get? What'd you get, Thomas?
Base substitution, missense, transition.
Nope. Nope.
Transversion.
Deletion.
What? I didn't look at that.
Right here, you'll notice
Yeah.
we've lost an entire codon. If you follow, everything's exactly the same.
So, what is it called?
What would you call this? A deletion.
Just deletion?
Yeah. Is this a frame shift?
No.
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 This is not a frame shift. The reason is you'll notice everything downstream is exactly the same.
No, I'm trying to figure out.
Yeah. So in this case, this was a weird one. We just call it a deletion. Here's an example of a deletion that's not a frame shift.
So it wouldn't be a base substitution.
It wouldn't be a base substitution.
It would just be
Yep, just a deletion. Isn't that funky? All right, let's look at hemochromatosis. What do we get?
base substitution, missense, transversion.
Transversion, that's exactly right. So base substitution, we have a G here. It's been substituted with an A. It's a missense because we go from cysteine to tyrosine. Right? And we're going from a purine to another purine. This is a transition. Yep. Purine to a purine, a transition. So base substitution, missense, transition. Any questions on that one? Okay. Let's go and look at our thalassemia, our first variant. What did we get? What'd you got, Thomas? That's right.
Nonsense, transversion.
Actually, on that one, all we have to say is base substitution nonsense. All right. The transversion transition only occurs with missenses. Only with missenses. Now, the cool thing about it is here you see, wow, you know, everything's exactly the same. But here we have a CAG, and here we have a TAG, and everything downstream is exactly the same. So that's just a base substitution. We go from glutamine to a stop codon. So that's a nonsense mutation. So we call a base substitution nonsense mutation. Any questions on that one? Let's go and do the last one, which I always think is the trickiest. What do you think, Thomas?
The base substitution frame shifter?
No.
No.
Not a base substitution.
Addition?
Not an addition. Now let me show you how I do this one. If you go and you start looking and say, okay, those are the same, those are the same, those are the same. Whoops. Now if it was base substitution, everything downstream would be exactly the same. But it's not. Notice how everything changed? And you say, oh, there must be an addition or deletion somewhere. So let's go where the change began. So if we look here and we look here, you'll notice that here's GTC and there's GTC. And there's TGC and there's TGC. Ah, those two A's are gone. That's a deletion. It is a frame shift. Yep, we call this a deletion frame shift mutation. Don't worry about putting missense in because it happens anyway. So we can call this deletion frame shift mutation. Beautiful. Now, we're going to talk a little bit about those mutations. Mutations are quite common. But we can generally say that most changes in the DNA are not going to help the organism and they're not going to harm the organism. We say that most mutations are going to be neutral. And there's several reasons why. So let me show you a little graphic with the reasons here. As I mentioned before, when we think about genes, we usually think about proteins. Right? And most genes for proteins only make up about a quarter of the genome. In fact, in this little chart, you can see it right here. These two parts here, this little pi and this little pi right here, that's all the protein coding genes. Right there. That's all the protein coding genes. The rest of it really has nothing to do with coding for proteins or RNA. Okay? The other important thing is, remember, in a gene, not everything is translated. A lot of stuff is spliced out. So if we look at the human genome, we find that 26% versus 2%, right, is going to be introns, and they're spliced out anyway. So if you have a mutation in an intron, that's not going to do a thing. Not going to do a thing. Right? If you have a mutation in one of these, not going to do a thing. If you have a mutation over here, it's not going to do a thing. Right? You have a mutation here in heterochromatin, probably not do a thing. Right? Even if you do have a mutation within the protein coding genes, right? Think about 1 out of 3 of those bases. That third base has a lot of wobble. Right? Because you can have many different codons coding for the same amino acid, variability in that third base will often go and give you the same amino acid. So a lot of them are silent mutations. Or if we have a little change here, it may not change the conformation of the protein or the shape of the protein enough to alter it into something terrible. So most mutations, because of the size of the genome, really make not much difference. Okay? Don't make much difference. So most mutations that occur are not going to help or hurt the organism. Okay, now we're going to talk a little bit about another level of mutations. We've been talking about point mutations that just affect a few bases. Now we're going to look at mutations that affect large regions of a chromosome, and these are called chromosomal mutations. And we use some different terminology for them. And we're going to start off with one that is called a deletion. Right? But instead of a few bases being deleted, it's a whole segment of a chromosome. Now, there are a lot of disorders that have this, But here's a famous one. This is called cry-do-chat. And as you can see, a deletion on chromosome number 5. Now, when you look at a chromosome, you might notice that there's a skinny part. That's right in the middle. That's a little constriction called a centromere. And you'll notice that you have a long arm and a short arm. So there's our short arm. We always call the short arm the P arm, and the long arm the Q arm. So as you can see, in this particular syndrome, we've lost part of the P arm on chromosome number 5. Now, this is an interesting disorder. Babies with cry-do-chat, in fact, cry-do-chat translates from French into cry of the cat, are usually small at birth and may have respiratory problems. Often the larynx, that's your voice box, doesn't develop correctly, which causes the signature cat-like cry. People with cry-do-chat have very distinctive features. You see her? They may have a small head, an usually round face, a small chin, widely set eyes, small bridge of the nose. Several problems occur inside the body as well. A small number of children have heart defects, muscular or skeletal problems, hearing or sight problems, or poor muscle tone. As they grow, people with cry-do-chat usually have difficulty walking and talking correctly. They may have behavior problems such as hyperactivity or aggression and severe mental retardation. If no major organ defects or other critical medical conditions exist, life expectancy is normal. So, this is an example of deletion where we have a big piece of the chromosome lost. And usually that spells bad news. Now, we have 20,000 different genes. All right? And one big question that biologists have asked for a long time is, where do genes come from? How do new genes form? Well, it turns out that question can be answered by a really common and really cool type of chromosomal mutation. A really common mechanism for creating a new gene is a process called duplication. In duplication, what happens is a segment of the chromosome makes a copy of itself, an extra copy, and may put that right next door. In fact, here's a type of duplication that's called a tandem duplication. Here's the original, and then the original makes a copy of itself, and now you have two identical sequences right next to each other. This is amazing. All right? So now you have the original, and now you have a backup. All right? If you have a backup, you now have something you can experiment with. Right? Oftentimes in films, for example, I read that during Harry Potter, Daniel Radcliffe went through probably about 30 different wands because he would just sit there and break them. He'd sit there and play drums, beat on them, and they'd break, and they'd have to get another one. All right. He would have been much more careful if they said, we got one wand, that's all we got. You know, that's it. All right. In the same way with natural selection, if you have two genes, all right, they both do the same thing, all right, natural selection is going to be a little less strict because you have the original and you have a copy. And because of that, one of those can change, can have a mutation that might alter and give us a slightly different protein. Now, take that as an idea. So let's imagine that we have a gene that makes a copy of itself, and those genes start accumulating mutations. Right? And over time, all of a sudden, instead of making the same protein, they start making slightly different proteins. but oftentimes proteins that have the same type of function. So what you would expect is if you have duplications over time, right, to start to see genes that look very similar to each other, that look like they're related to each other. In fact, we find them all over the place. We call them gene families. And let me show you what we mean by a gene family. All right. These gene families are pretty cool. You can go and you can look through a variety of different organisms, and you can start to say, wow, that gene and that gene and that gene all have to do with DNA repair. But their sequences are really similar to each other. I wonder if they're related to each other. And then you find out, hey, they are. They're related to each other. And if you go and you find them in the same family, or the same order, or within the same kingdom, and you say, oh, that family must be really, really old. There must be a really distant ancestor that was the original gene, the rather founder gene for that particular family. Now, one of the cool things, if you go to the National Library of Medicine, you can look at this little genetic home reference. And as you can see, it tells you all the gene families that are found in humans. And as you can see, I've just gotten to the C's there. These are collections of genes that usually have a similar function. All right? The reason that they're similar is they're descended from an original gene that duplicated, and then later on that duplicated, and then later on that duplicated, and they each followed their own evolutionary history. Let me show you one. This one's pretty cool because it's really old. We've had DNA for at least 3.5 billion years. All right? So repairing DNA has to be pretty dang old. We have a whole family of genes that are just there for repairs. This is called the Rec family gene. The cool thing about it is here we have in humans all the members of this family, and they all have very similar sequences. But the cool thing about it is we find members of this family in distant related organisms. This is a plant. Here's one in E. coli bacteria. All right. This one is an archaean. All right. So this family is really, really old. Now, one of the most fascinating ones that we can find in us and other vertebrates is one that produces hemoglobin. In fact, hemoglobin, if you remember, has two proteins, the alpha chains and the beta chains. It turns out that the genes for those are actually found on two chromosomes, chromosome 11 and chromosome 16. And the cool thing about it is we find that they are members of gene families. So let's take a look at them. In fact, this is a cool picture here. On chromosome number 16, all right, if you look at the hemoglobin molecule, you have two alpha globins. The genes for making those are found on chromosome number 16. All right. The beta globin that we've seen before, those are all made from genes on chromosome number 11. And so if you look at the chromosomes, you'll find, hey, all these are related to each other. Right? All these alpha globin genes. Now, the interesting thing about them is some of them are expressed at different times in development. For example, this alpha globin, this version, which is a slightly different protein, is expressed during our embryonic stage. These two are expressed when we're both a fetus and an adult. In the beta globin, you can see we have one for the embryo, one for the fetus, and one for the adult. All right. One other interesting thing is you'll notice too that we have these strange ones. The ones in green, those are bizarre. What you're seeing are what are called pseudogenes. Pseudogenes are almost like fossil remnants of genes. These are genes that were once working and viable until they got knocked out by a catastrophic mutation. But we still have them in us. So here are a couple pseudogenes. You can see we have 4 of them, all right, that once coded for a globin, but no longer function. But they're still there. It's almost like going and finding, you know, an ancient artifact from some distant civilization that no longer exists, but existed at one time. And this is in our DNA. If you look here, we also have a beautiful little pseudogene that's found in our globin. These pseudogenes are absolutely fascinating, and they can be produced through this process of duplication. So here we have a gene. We get duplication, so we have 2 copies of it. They're both being subjected to mutations, and one just happens to have a catastrophic mutation that knocks it out. But it's still there. It's still there in the population. Right? So I like to think about pseudogenes as defunct relatives of genes. Defunct relatives of genes. And they have some really, really cool examples. Right? Now, we have a lot in humans. This is actually from an organization that tracks the pseudogenes in humans. This is pseudogene.org. And as you can see, we have identified 8,000 plus 4,000, 12,000 pseudogenes that are found in the human genome. The cool thing about them is that these genes might be functional in relatives. I'll show you a couple of examples of one, but the last thing I want to do is show you a little video that looks at a really cool pseudogene in a fish that lives in Antarctica. So a really cool example of the formation of a pseudogene. So let me go ahead and